So next up, we have Mike Sapieha from the University of Montreal in Canada, and he's going to talk to us about innate immune memory in AMD.
Great. Well, first of all, thank you very much to all the organizers, Sarah, Florian, and John, for the invitation and giving me the opportunity to speak about a program that we've been developing and that I think a biology that is getting more and more exciting as the community collects data in different animal models and different clinical settings. Yet nothing in AMD so far in terms of clinical data. So I do have these little disclosures to announce, and I'll move to Diane. I changed the outline of the slides, and I actually put a tree here. My lab works on aging. This is the Macular tree. It's the oldest living organism on the planet. It has only an innate immune system, so keep that in mind. And the questions that I'll try to answer today is really, how does the immune system influence AMD? What's the impact of something called trained immunity on AMD? And can we therapeutically modulate immunity for AMD? So I'll start off with a little bit of an anecdote, and I'll go back to this fellow in a couple of slides. This is Elie Metchnikoff. He's considered to be the father of innate immunity, but more importantly, he's also been given the title of the father of gerontology. And so he discovered or coined the term phagocytosis or phagocytes, and essentially towards the later part of his career, he was interested in understanding how we age and why do our tissues essentially degenerate with age. And of course, being centered on macrophages and microglia, he noticed a lot of inclusions in older tissues versus younger tissues. So he came up with the concept that devouring phagocytes are the culprits of senescence. And by senescence here, he means aging. And just two other little things that I thought was interesting to mention and that I learned by reading about Metchnikoff is one that after his first wife died from tuberculosis, he attempted suicide by taking a very large dose of opium. And fortunately, he didn't succeed with this. So when his second wife got quite ill with typhoid fever, his own heart and eyesight were failing. And so he attempted suicide a second time. And this time he did it in a bit more of a scientific manner. He started injecting himself or inoculating himself with higher and higher doses of relapsing fever bacteria. And surprisingly, what he reported was that his eyesight actually got quite a bit better after this. And also his heart condition apparently resolved itself. So I thought this was kind of an interesting concept, because I'll be talking to you for the rest of the time about how we can tweak our immune system to maybe favor outcomes in age-related eye disease. So since Metchnikoff, there's obviously been a lot of effort to try to understand what happens with your inflammatory system as we age. And so there's been several epidemiological studies. One that was particularly well done is this aging in the Chianti area. So this is an area of Tuscany where people live longer than in most other areas. And in 1998, they analyzed 1,270 patients, and they came up with these blood cytokine levels. So you can see here that in both men and women, there's a significant rise, primarily in IL-6, but also if you look at the bottom part, in C-reactive protein that Robert spoke about this morning, and other ones like IL-1 receptor agonist. Although when you actually do regression analysis, and I'm not showing that data here, it normalizes quite a bit to a baseline level if you take away the factors of heart disease and morbidity. So what this actually means is that while our levels of systemic cytokines rise with age, the fact that we have morbidity or cardiovascular events might contribute to this rise. Or the opposite, that this rise in cytokines actually contributes to morbidity and cardiovascular events, and that relationship is not really clear yet. So if you take the same data, so the Innate trial that you see here in red, and compare it to other epidemiological studies that were performed around that same period and more recently, and you analyze all of those curves of cytokine increase simultaneously, you get something that is quite interesting. So essentially, populations that are from non-industrialized countries, as the one here that you see that comes from Bolivia, and this other one that comes from a agricultural part of Singapore, you see that this increase is actually quite flat or flattened. So they have much higher levels of baseline inflammation. And maybe what's even more interesting is that their higher levels or their baseline levels of inflammation don't actually correlate with morbidity. So whereas in the Italian group and the other groups, you have a big correlation with diseases such as kidney disease and diabetes. This is completely absent in those populations. So the idea there being that the non-industrialized populations are exposed to bacterium throughout their life and infections and so on. So they in a sense prime their immune systems, and essentially there's no more correlation between higher inflammation and these diseases of aging. So one of the concepts that I'll be talking about is trained immunity. So the immunology as a discipline was really born after Metchnikoff that we spoke about in the first slide, and Paul Ehrlich were given the Nobel Prize in 1908 in physiology and medicine. So Metchnikoff being the one who discovered the innate immune system, or discovered the processes that led to the concept of the innate immune system. Paul Ehrlich being the one that described the side chain theory of antibody formation, and essentially is considered to be the father of adaptive immunity. And so this led to over the next century, the mapping out of different cell populations that fall into both branches of the immune system. So the innate immune system is thought to be the first barrier. It's also thought to be quite slow. Quite rapid, and non-specific. So essentially works through pattern recognition receptors. So everything that the immune system detects as non-self will go and attack. And it's also classically thought not to have a memory. And the adaptive immune system, to the contrary, evolved much later, and it's centered on lymphocytes, so T cells, B cells, and it's generates antibodies through somatic combination. So it's thought to be slow. It takes several days to kick in, highly specific, and it has this memory that you can see with different types of vaccinations. So if you were to look at this, so if you're exposed to a pathogen or damage, then your T cells and B cells interact together. You get clonal expansion. Then you have memory T cells and memory B cells, and they act in a very selective manner. So this is the kind of classic vision of our immune system. Now, around 90 to 97% of total biodiversity on Earth is devoid of an adaptive immune system, yet it's survived really well. So over the last 15 years, there's been this concept that immune memory is not just restricted to the adaptive system. It's actually probably something that's also inherent to the innate immune system. And so the way this would work is that if your innate immune cells get exposed to stressor, they reprogram their DNA landscapes to make them ready for the next stressor. So essentially you can prime your immune system, reprogram it, and when you have a secondary infection or secondary presentation of a pathogen, your immune system is much more ready to act on that. So this would be a form of innate immune memory or trained immunity. And so the way this works, you can see here you have a resting state innate immune cell, a monocyte perhaps, or a macrophage, so nice condensed DNA. Then you get your first stressor, you're ready to produce cytokines, so you get a bit of remodeling, so you get acetylation of certain histones, and then when you go back to a resting state, you don't go back to your initial resting state because you've already marked some of your histones for rapid response next time. So when you get your second hit, then you're really ready much more quickly to respond, or to a greater extent to that same or parallel stressor. Now, maybe some of the most compelling evidence that this idea exists comes from the BCG vaccine. So the BCG vaccine was essentially developed in the early 1900s by Calmette and Guérin. And essentially what they did was they used a live attenuated form of Mycobacterium bovis, so a cow form of tuberculosis. And they attenuated this virus. They mixed it with different components of ox serum, and eventually this became the most widely used vaccine in the world. So it's thought that around 130 million children get vaccinated with this vaccine every year. And interestingly, in Europe in the early 1900s, they noticed that this BCG vaccination reduced very significantly the amount of childhood mortality. And this cannot account just for tuberculosis. So there was some sort of cross-reactivity happening. And more recently, randomized clinical trials showed that up to 50% reduction in mortality in infants that were of low birth weight that were vaccinated with BCG. And there's other evidence that if you're vaccinated against BCG, you can be protected by completely independent pathogens such as RSV, influenza A, herpes simplex, and maybe the best known case in oncology is bladder cancer. So being vaccinated with BCG confers significant protection for bladder cancer. And what we now know is that this BCG virus can actually reprogram your stem cells in your bone marrow, so the ones that are responsible for myelopoiesis. And essentially they're released and they're activated and ready to combat these parallel infections. So the immune system in AMD is something that's gained a lot of interest over the past 2.5 decades. And essentially one of the first studies that really proved that you can target the innate immune system, or that an anti-inflammatory drug could work, was published last year in JAMA Ophthalmology by the group of Anneke den Hollander. And what they found was that essentially patients that had autoimmune disease and that were already on JAK-STAT inhibitors had a significantly lower rate of incidence of both dry and wet AMD. So if you look at the actual incidence curves, you get somewhere around a 43% reduction in incidence in one of the cohorts that were analyzed, and somewhere around a 70% reduction in incidence in the other one. So really proving that if you modulate your immune system, you might be able to slow down or lower the incidence of AMD. Now, the work that was that really kind of put the concept on the map was done by Florian Senlaub's lab, and essentially what they showed was that as you get progression from a healthy retina to small drusen to large drusen, there's an accumulation of an immune cell population called mononuclear phagocytes, and essentially you can really see them co-localizing or congregating around zones of atrophy, and eventually, if this doesn't resolve, then you can convert to a wet AMD phenotype. Now, other work also from the Paris Eye Institute. So these are time-lapse flood imaging, and you can see these hyperreflective foci that congregate around drusen, and you can see them really moving around zones of atrophy. So here you see at the border region, and there's really this congregation of the hyperreflective foci. And also Florian's group later showed that these hyperreflective foci have all the markers that you would see in a macrophage. So they're highly mobile, but if you look at, if you profile their surfaceomes, they are essentially macrophages that are moving around. So with that, there's other work with done by OCT here by Michel Pak, and he followed, this is a 5-year time lapse, and you can see this hyperreflective foci that's always at the border of this, I guess, reticular drusen that's advancing. And the last evidence I'll show is actually not in an AMD model, but in a model of retinal degeneration done by Wei Wang's lab when he was at the Wai-T Wang's lab when he was at the NIH, and he showed very nicely that microglia in this case could engulf and eat up photoreceptors. So we spoke about the concept of innate immune memory, and is there an impact on progression of age-related eye disease? So essentially the way we're looking at it is that microglia, or mononuclear phagocytes, throughout life are you're exposed, and they're exposed to viruses, to bacterium, and to Western diets. And this can potentially reprogram your innate immune system and drive disease. And so some of these innate immune cells can live several years in your CNS. And so hence, if you reprogram them at one stage of life, there's a possibility that this aggressive phenotype that's initially there to protect against future infection or to deal with lipid overload can actually cause issues and contribute to retinal degeneration in AMDs. And so to get an idea a bit of how long a microglia can live in your CNS, there was this very clever study done using C14 derived from atmospheric C14 derived from nuclear bomb tests over the South Pacific. And so if you look at environmental C14 levels, you can see that they really spiked around the late 60s when a lot of the nuclear testing was being done, and they're now subsiding, and hopefully this will go down to zero shortly. And what you can do from there is you can essentially do a pulse chase experiment where you can date the cells within your body. So the conclusions of the study showed that on average a microglia can live 4.5 or 4.2 years, but there are populations of microglial cells that can go up to 2 decades or more. So essentially they live longer than we initially thought in the CNS. And so we wanted to see if any of the environmental factors that predispose to AMD could trigger or reprogram some of these microglia or some of these innate immune cells that you might eventually find in your CNS. And so Tiernan this morning went through, very exhaustively and very elegantly through the risk factors that predispose to AMD. The one that we were particularly interested in were the metabolic risk factors. And although, the jury's out on exactly how important they are for driving AMD, there were some compelling studies that showed that an increase in men of your waist-hip ratio by an odds of 0.1 led to 13% increase in AMD, in early AMD, and up to 75% in late AMD. So again, at least in some population studies, the BMI seems to be an important risk factor for development of AMD. So how does past metabolic stress apply, or how does it influence development of AMD-like lesions? So we used a mouse model where we took mice that were 8 weeks old and then fed them either a regular diet or a high-fat diet. And so this eventually, after a couple of weeks, these mice become obese, and then we take them off the high-fat diet 11 weeks after we put them on. And then we wait another couple of months until you can essentially not distinguish whether these mice are obese or not. So phenotypically, they look exactly the same. And also, when you look at their blood work, so whether it's their insulin levels, their insulin response, their ability to control glucose levels, they're essentially indistinguishable from healthy mice. So we took those mice and then used a model called the laser-induced CNV model that is, pretty much the gold standard for neovascular AMD work, although it's got many caveats. One of the issues here is it's a highly inflammation-driven model. So you do see some of these features in human pathology, but it's very staged. And at one point, the actual monocytes can start eating up the neovascular lesions. So, something that you have to essentially apply with different other models to really have confidence that what you're seeing in the CNV model translates or can translate to a clinical setting. So with this model, what we found was that the mice that had previously been obese actually developed much larger CNV lesions. So they had a much higher angiogenic or pathological angiogenic burden than the mice that were always slim or always on a regular diet. So you can see this significant boost in those mice. Now, interestingly, when we actually looked at the number of immune cells in those retinas, they didn't vary. So there was the same amount of immune cells in the mice that were previously obese and now no longer obese than the ones that were never obese. So we then took adipose tissue, so directly from the mice that were previously obese, and transplanted them onto mice that were not obese. And we also took mice that weren't obese as controls and transplanted adipose tissue from non-obese mice. And again, we're able to recapitulate this phenotype where mice that were previously obese could confer some sort of memory to the skinny mice where they developed more CNV lesions or bigger CNV lesions. And once again, the number of immune cells stayed the same. So we have the same amount of immune cells, but they're in a different type of state. And so when we looked at the weight of the adipose tissue pads, of the fat pads, we saw that the ones that had returned to regular diet were essentially indistinguishable from the ones that were regular diet, so the ones that were always metabolically healthy. And the one thing that we did find was mononuclear phagocytes that were positive for CD38. So this is a sign to us essentially of an activated immune cell. So we had these long-lived activated immune cells in those fat pads. And when we did another experiment where we took bone marrow from these mice that were previously obese and transferred them to regular mice, we also found this increase in neovascularization. So really suggesting that the whole mouse was essentially had monocytes that were primed for producing pathological neovascularization. So how exactly, what are these changes that we were what were the changes on the level of their DNA landscape that might be driving this phenotype? So we used a technique called attack-seq. And essentially here what we do is we look at what parts of the chromatin are open, and we do sequencing, and we come up with maps of which parts of the chromatin in an activated or previously obese mouse are more open versus the ones in a regular diet mouse. And what we can see right away, if you look at the principal component analysis, so you have the mice that were on high-fat diet, they cluster differently. And then you have the mice that were returning back to regular diet that cluster back together. But when you look at the actual expression of or the differentially regulated chromatin, the differentially regulated genes, you could see that the mice that were previously on the mice that were on a regular diet always really differentiated significantly from the mice that had returned. And essentially, mice that had returned on a regular diet were indistinguishable from the mice that were still on a high-fat diet. So what this means is that you have whether you're still obese or whether you've lost the weight and now you're metabolically healthy, there's the DNA signature is essentially identical between the obese mice and the ones that had previously been obese but returned to a regular healthy state. So with this, when we started looking at what genes were regulated, we saw that the primarily pathways for inflammation or angiogenesis were being opened. So the zones of chromatin that were the most open were really the ones that were coding for pro-angiogenic phenotypes and pro-inflammatory phenotypes. So we then wanted to see if we could recapitulate this in an in vitro setting, and so we first ran analysis to see what lipids were the most regulated during obesity. And what we found was that stearic acid was the most highly regulated. We could recapitulate this phenotype to a lesser extent with palmitate, and then when we treated the mice, bone marrow derived monocytes, we were able to see increases in inflammatory cytokines. So really thinking that these lipids are the culprits for reprogramming the monocytes. And also what those lipids were doing were they're conferring a higher glycolytic capacity to the monocytes. And essentially, the more a monocyte has a glycolytic capacity, the more it shifts towards glycolysis, the more it's ready to produce cytokines. So all this together really suggested that we had immune cells that were either in the adipose tissue or circulating or perhaps in the retina that were reprogrammed and acting in an aggressive manner in these healthy mice as much as they would be if the mouse had always been obese. So then we did two last experiments here where we took a mouse where we could eliminate the mononuclear phagocytes directly from the adipose tissue. And when we eliminated the monocytes from the adipose tissue, we were no longer seeing any difference between the phenotypes. So essentially, there was a program in the adipose tissue macrophages that was driving this phenotype in the distal tissue of the retina. Now, we did the same thing, but this time we got rid of the microglia in the retina and really came to the exact same phenotype. So we're still trying to figure out how this transfer from adipose tissue macrophages and this recapitulation by microglia works. But what we did notice is that some of these adipose tissue macrophages were actually migrating towards the retina. So this is ongoing work that we're trying to figure out. Now, another thing that we looked at was how does a past history of infection impact immune behavior in the retina? And so here we were inspired by work done by Joan Miller's lab, where she noticed that patients that had AMD and neovascular lesions also had IgGs against chlamydia pneumonia, so the lung infection, not the venereal disease. And essentially, you can see some of these bacillus of chlamydia inside of the neovascular lesions. So what we did was we then inoculated our mice with chlamydia and waited several weeks, several months, so their baseline inflammation dropped down to normal. Yet they still, when we induced the neovascular lesions, they were much larger in the mice that had previously been been exposed to the chlamydia. So again, much like the mice that were previously obese, these mice seem to have a type of immune memory. So there's something with the layout here. I'll go quickly through this. So essentially, with these experiments, what we did was we also used the mice where we could eliminate either local or systemic monocytes in the retina, and again found that eliminating the local microglia would be enough to return these activated mice back to a regular state, so one that was devoid of infection. So lastly, to find out if this is a druggable mechanism, we wanted to see what processes were at play and can we interfere with them to alter the course or this aggressive neovascular phenotype. So we did some attack-seq on single-cell attack-seq on retinas of mice that had received the training paradigm, this time with LPS. And unfortunately, something happened with my slides. But you can see here in C3, this is a unique population of microglial cells that we isolated that when we look at, they're unique to this training paradigm. So essentially, we had 6 different types of immune cells that we were identifying with the single-cell RNA-seq, and only the C3 was present when we had primed the mice with LPS. And then when we looked at what parts of their genome was being most regulated, we found that transcription factors, including ATF3, which is part of a larger family called activator protein 1, so AP1, were selectively regulated. Now, when we looked in the obesity and regular diet model, we found also that the top 10 most regulated genes, or top 9 most regulated genes, were sites of landing for transcription factors, not genes, were also for AP1 family members. So we thought this was kind of exciting because both of these training paradigms converged on the AP1 family, and we confirmed this using an approach called ChIP-seq. So the AP1 family is a very broad family. It's got at least 20 to 30 different subunits. And so we've been trying to see if we could have an oral compound that can target these pathways. And we have a tool compound that we call MT1. And essentially MT1, when we give it orally to mice, it's able to very significantly engage its target. So we look at AP1 binding to different other partners, and you can see that with an oral dose, this AP1 is dissociated essentially, so it's no longer able to function. We also have good mechanism engagement where we give an oral dose of MT1 and then induce an LPS stress, so an immune stress, and collect the brains of these mice, and we see a significant drop in cytokines within these brains. So we then put this in a model of photoreceptor degeneration, so light-induced photoreceptor degeneration, and found that we can reduce the amount of photoreceptor degeneration with oral treatment of this with MT1 by at least 40%. So this is work in development that we're trying to see the viability of this approach, but certainly we're excited to see that by modulating these pathways, we can recapitulate, or we could salvage the outer retina. So as a summary, we found that 2 different approaches, so either one that confers metabolic stress to the mouse, or one through an infection, that we get reprogramming of the innate immune system, and that this innate immune system is able to act on distal organs specifically of the retina, and exacerbate CNV lesions. So, as a summary, we think that past history of obesity, whether it's so essentially if there was obesity at an earlier stage of life, or systemic infections, that they can impact progression of AMD in mice, or of CNV in mice. Then by interfering with these pathways, or with other means of interfering with systemic inflammation that's targeted, for example, the JAK-STAT pathway, we can also impact the incidence of AMD. And lastly, I think investigating this concept of trained immunity can potentially provide some input into druggable pathways for AMD. And with that, just the unanswered questions that remain. So, how well can we really drug this? Is it better to reprogram these cells that have undergone trained immunity, or is it better to eliminate them? Is this a concept that's relevant to other eye diseases? And is this laser model that I've been showing you actually relevant to extrapolate findings to neovascular AMD? And just for further reading. Thank you.
Oh God, lots of questions. Why did Mechnikov see better after his infection?
Yeah, good question. I don't...
Hi, Lindsay Chu, MD PhD student at Duke. Thank you so much for your talk. I was wondering if, maybe I just missed it, but I was wondering about any ERG data to correlate with like the visual function findings that you were mentioning for the CNV model. And then I was also curious if you have thought about or have previously explored not just starting the high-fat diet at the 8-week time point, but in even younger mice, since childhood obesity is also a major issue in the developed world and even the developing world, and thinking about how that impacts these kinds of findings.
Okay, so those are two great questions. So the second one, so we've not looked earlier on, but I think to your point, there's some very strong epidemiological data linking childhood, so prepubescent obesity, to multiple sclerosis, and also adolescent obesity to multiple sclerosis. So I think to your point, going earlier, although at this point they're juvenile, so I don't know if we would really see anything more exacerbated than what we see here. Now, in terms of ERGs, we did try ERGs. So in this CNV model, you're destroying parts of the retina. So getting actual better ERG signals, was maybe not necessarily the relevant readout for function here.
Hello. Thank you for a wonderful talk. I'm very excited about you finding epigenome modulation by diet. I was wondering, because in different tissues in the brain, it has been shown that caloric restriction also can change the epigenome. Have you considered kind of priming perhaps microglia with a healthier diet or just caloric restriction, and see if that affects the progression of some AMD phenotype?
Yes, so we have not done that, but certainly a very compelling idea. So I think we did look at intermittent fasting. So the classic 16-8 hour time frame, and we wanted to see if we could reproduce or kind of palliate the juvenile obesity. And what we found was in that particular model, so it's not caloric restriction, but intermittent fasting, we found that the mice actually got a lot fatter, the ones that were caloric or on that 8-16 paradigm. So we've not looked further into that, but certainly a very interesting idea.
Thank you.
Question was answered. Yeah.
The question was answered.
I was about to ask about time-restricted feeding, but
Yeah, okay.
Actually,
I have a question in that case, maybe. Have you tried a different model other than the laser-induced CNV for your obesity model? Like, have you just even looked to see if, if you age out the animals, whether you start to see infiltration of cells?
Yeah, so we did do the light-induced photodegeneration model, and we did see significant gains in that, well, significant protection in that model. Yeah, so at least in those two models, we're seeing significant benefits.
So that's with your MT1?
No, but we also did it in the obesity model. I didn't present the data, but we did that, and it's published. Yeah.
So when if I just eradicate all my resident macrophages with the MCSF receptor inhibitor, will I lose the innate memory and be all fresh, or does that work?
So that's a good question, and likely yes, because I mean we're using a much more, I guess, penetrant version of that, right? Where we're using a diphtheria toxin driven by CX3CR1, so you would expect, I guess, the ultimate outcome to be quite similar. So either doing that or using a LISM driver line that will hit myeloid cells, you can salvage that phenotype.
Thank you very much. Fascinating talk. So in human AMD, we've never ever managed to find a really good link between AMD severity and body mass index or any of the other markers that we've used. We've looked at cholesterol, we've also looked at triglycerides, we've tried all these different features of dyslipidemia and never shown it. And I'm just wondering, have you used human biological samples, people who have very severe AMD, and to see whether this might influence your animal models in any way? Since you're considering transplanting other biological material into animals.
Yeah, it's a very good question, good point. So, yeah, I think initially we were pretty excited about the BMI studies that were coming out of Australia, and but yes, we're aware that that field is conflicted, and Tiernan spoke about that this morning. So I think what we see in our data, whether it applies to AMD, is still to be determined. I think what is clear is that you have a primed immune system that's ready to act in a different manner than it would in an otherwise healthy individual. And whether this translates directly to neovascular lesions, that's to be seen. But certainly, again, this the link between childhood obesity and neurodegenerative disease, or specifically MS, is there. So I think, again, not fully on for AMD, but I think you are reprogramming your innate immune system, and if it's involved in AMD, you would expect that there would be a contribution.
I have a question. Sorry, over here. Oh, good. Okay. So in your obesity studies, have you tried to extend the time between coming off the high-fat diet and giving the CNV to see whether there's like a threshold for like how long they hold on to this memory?
Yeah, that's a great question and a very long experiment. So we've done it actually up to a year, and so we saw that there was still that memory that remained, but it wasn't as pronounced as when we did it in 3 weeks. Now there's some literature that claims that you can have transgenerational transmission of this. It's been a bit disproved by other literature. So certainly, features. I mean, if you think about the studies that looked at the Great Dutch Famine, 44 to 45, or the Great Chinese Famine, you see a lot more obese offspring in those cohorts, and that can be transmitted 2 generations. But with the actual immune protection, it's really not clear. Especially if we think it goes to myelopoiesis, that's not clear that would be transferred to the next generation.
Mike, beautiful work.
Okay.
Extrapolating your talk into Matthew's, or maybe other way around, have you looked at the diurnal increase in innate, systemic innate immune cells in the retina?
Yeah, that's a great question. We've not done that, but certainly would play into this, right? And especially depending on what time of the day we do our models.
Nishan Ghosh from Johns Hopkins University. Very nice talk. My question is that when you're talking about the innate memory, can you also comment that what really happens when you, the status of the resolution factors in the system, particularly resolvins and all those molecules, and does it impact on the state of the immune cells in the system, in the mouse?
Okay, so that's an interesting question. So whether you have a compromised resolving capacity of these cells. So we've not looked at resolvins or neuroprotectants or any of those DHA byproducts or EPA byproducts. What I can tell you is that we have the exact same number of mononuclear phagocytes or of microglia or any other immune cell that we looked at pre and post lesion. So it doesn't seem to really influence movement or resolution, but really the cells are there and they're just in a different primed state.
Thank you so much.
Okay, thank you very much.